Have you ever wondered how scientists study specific proteins in a complex mixture? The answer lies in a powerful technique called Western blotting. Let's uncover its history, procedure, advantages, and limitations. Western blotting was first introduced in 1979 by scientists Harry Tobin, Joe Gordon, and Julian Gordon. The name Western is a playful nod to the Southern blot, a DNA detection method developed earlier. While the Southern blot focuses on DNA, the Western blot focuses on proteins, opening up a new frontier in molecular biology. So what is Western blotting? It's a laboratory technique used to detect specific proteins in a sample. By separating, transferring, and labeling proteins, we gain insights into their presence, size, and abundance. First, we extract proteins from a sample, often using a lysis buffer to break open cells and release their contents. Proteins are then quantified to ensure consistent analysis. But much of this process should be taking 2 or even 5 times longer. So one would expect that at least half or more of the proteins can be broken apart and then released. Next, proteins are separated by size using SDS page. Smaller proteins move faster through the gel, while larger ones lag behind. This creates a size-based separation. The separated proteins are then transferred onto a membrane, typically made of nitrocellulose or PVDF. This step preserves their arrangement for detection. Blocking the membrane prevents non-specific binding, ensuring that antibodies stick only to their target proteins. A primary antibody is added to bind to the specific protein of interest. Then, a secondary antibody linked to a detection marker binds to the primary antibody. Finally, we detect the protein using techniques like PF chemoluminescence, fluorescence, or colorimetric assays. The result is a visible signal, often represented as bands. To begin, Western blotting has several strengths. It is highly specific, able to distinguish between similar proteins. It is versatile, allowing detection in diverse samples, and provides quantitative insights when combined with modern imaging tools. However, Western blotting has its challenges. The procedure is time-intensive, requiring careful handling. It also demands technical expertise and can be costly, especially for advanced reagents. Any thoughts? I'll keep in mind. Despite its limitations, Western blotting is invaluable. It's widely used in research to study proteins, in medicine to diagnose diseases like HIV, and even in forensic science for evidence analysis. As science advances, Western blotting continues to evolve. Automation, enhanced detection methods, and integration with other technologies are making it faster and more precise. In summary, Western blotting is a cornerstone of protein analysis. Its ability to provide detailed insights has revolutionized fields from medicine to molecular biology. So, the next time you hear about protein research, remember the Western blot, the silent hero behind countless discoveries. Thank you for watching, and happy exploring the world of proteins.